Hi, everyone. It's Dr. Sheftal again with Truth in Science. Today, we're going to have hopefully a very interesting presentation. It involves some of the risks that you undergo when you have the messenger RNA vaccines. And we're going to talk about some of the risks, and then we're going to hopefully get an expert. I'm going to try to show a video of an expert talking about one of these risks, and then we're going to analyze what he said a little bit. But I've never done a video on one of these things before, and I hope I can do it. So let's get started and see if I can pull it off. Okay, this is the bona fides page, which we always show and which I will skip as usual. Okay, just to review what happened in part one, it was about the mathematics or the arithmetic, if you will, of these messenger RNA vaccines. I strongly urge you to go back and look at part one if you have not seen it yet. It explains everything in detail, and you'll be surprised at what these numbers mean. So I do urge you to do that. Today, again, we're not going to talk about the numbers. We're going to talk about some of the risks involved in getting the vaccines. And this is a good time for me to say that I'm not pushing people to have the vaccine or not to have the vaccine. I'm just trying to help people understand the implications of the numbers from the raw data that Pfizer provided and Moderna, and also to talk about some of the risks associated with having the vaccines. I'm not trying to tell people to have the vaccine or not have it. So let's proceed and talk about the risks today. All right, first, the first type of risk that I want to talk about is if some of the genetic material from the vaccine gets incorporated into your own genetic material. And at first, I thought, well, it's messenger RNA, and that translation occurs in the cytoplasm, and it would be very unlikely for this messenger RNA to get incorporated into your DNA. It would have to get converted to DNA by reverse transcriptase first, and then it would have to find itself in the nucleus where it could possibly interact with your DNA in some way where it could be incorporated. But then I read two peer-reviewed papers, and let me just read what one of them said here. It says the genomic integration of DNA vaccines into host chromosome is another biosafety concern, which may lead to mutagenesis and oncogenesis. Oncogenesis would mean birth of cancer, for example. Even though previous studies have shown that the risk of vaccine plasmid insertion into the host chromosome is pretty low, the FDA and the WHO still recommend integration studies be included as part of the safety program of DNA vaccines. So they're saying that the risk of vaccine plasmid, that's the genetic material in the vaccine, the risk of vaccine plasmid insertion into the host chromosome is pretty low. That's a little scary, isn't it? What do they mean by pretty low? Okay, and then the next quote involves viral vector vaccines, such as the AstraZeneca and the Johnson & Johnson vaccine. And let me just read you this quote also. It says, viral vector vaccines are recombinant viruses that encode antigens of interest in an unrelated modified virus. So let me just decipher that for you for a second. We're taking a different virus and we're going to take a piece of the coronavirus a piece of the coronavirus's genetic material that codes for the protein you want to make antibodies for. In the case of these vaccines, it's the spike protein, okay? That's what recombinant means. It means just taking a piece of genetic material and inserting it into the genome of the vector virus, okay? It'll be clear when I show you the picture down here. AstraZeneca uses non-replicating human adenoviruses and the Johnson & Johnson virus uses a non-replicating primate adenovirus. Venezuelan equine encephalitis virus is the most commonly used for coronaviruses. Recombinant viruses carry the risk of integrating their genome into the human host, so additional biosafety assessment will be required before entering clinical trials. Again, a little bit scary there to hear that that's sort of a common thing, or it's a possible thing even, and additional biosafety assessment will be required. But let me just point out on this diagram here how this works. So you've got the coronavirus, and they're going to take a little piece of the RNA in this coronavirus that codes for the spike protein and insert it into a modified chimpanzee virus, which is depicted here in green. And there's the piece that codes for the spike protein. And then infect you basically with the modified chimpanzee virus so that its genome gets into our cell, and then we make the spike proteins and form antibodies to it. So it's similar to the messenger RNA vaccine, but instead of having the messenger RNA go directly into the human cell where it will be translated into the spike protein, we use a virus, in this case, a modified chimpanzee virus, to be the vector. In other words, the thing that goes up to the human cell and the cell walls meld together and it delivers the genetic material into the human cell. Kind of like the way coronavirus does so naturally, only they're going to use a modified chimpanzee virus to deliver the genetic material to the human cell. Okay, now the main thrust of, now that's the end of the risk of having the genome or the genetic material from the vaccine incorporated into the human genetic material. And now I want to talk about something called antibody-dependent enhancement. There are three mechanisms of antibody-dependent enhancement, which we'll call ADE from now on. The first mechanism occurs when there are not enough antibodies produced by a vaccine or a previous infection or whatever to clear the virus or neutralize it, so that a few antibodies stick to proteins on its surface, as is shown here. And if you remember the primer that I gave you on immunology and the first and longest over 4-hour video that I made, you'll remember that there were antigen binding parts to the antibodies and a constant portion called the FC portion of the antibody. And on the surface of the cell, the phagocytic cell, there are FC receptors, which are depicted by these red kind of goalpost shaped receptors here, which the FC portion, which is this portion here of the antibody, after it's attached to the virus, itself attaches to the red receptors. And then that helps the phagocytic cell phagocytize the virus, bring it into the cell, where it can, since it hasn't been neutralized, it can hijack the replication machinery in that cell and make copies of itself. So think about it. That's antibody-dependent enhancement, because that makes the virus more delectable to the phagocytic cell, which is a bad thing, because that virus, since hasn't been neutralized, is just going to hijack the machinery inside the phagocytic cell and make copies of itself. We certainly don't want that to happen. And I'm going to talk about dengue fever in just a minute. And this is one of the things that happens in dengue fever. Antibodies to a different type of dengue fever from a previous infection are still present in the human, in the person who was infected. And those antibodies are not specific or strong enough to neutralize the dengue fever that you get in the second infection with a different type. And the illness is much, much worse the second time because of this phenomenon here. Okay, it actually helps the virus replicate itself, which makes the illness worse. Now over here, just to compare, here's an example of a virus that was neutralized by many, many antibodies. And when it gets brought in, it's just been torn to pieces and it's unable to hijack the machinery and produce copies of itself. Okay, so that's in contrast to the incompletely neutralized virus and what it can do. And that's why this is enhancement, okay? The second mechanism by which antibody-dependent enhancement occurs is through immune complex formation. That's when antibodies attach to two viruses at the same time, and a big clump of antibody and virus forms, which causes all kinds of problems, for example, in the lung. Lots of cytokines get released, the lungs fill with fluid, complement cascade gets activated. And if you remember again from that first primer, I explained what complement was. And so this is dependent on antibodies, and it certainly enhances the damaging effects of infection to where it's much worse than it would be if the antibodies were not there from the previous infection or from a vaccine. And finally, the third type is depends on where the antibody attaches. Remember, this is the spike protein, okay? And there are a lot of epitopes on here, which are just antibody binding sites. They're antigens where the antibody attaches. And it turns out that the N-terminal domain, which is depicted in green here, if the antibody attaches here, it will enhance the receptor binding of the SARS II spike protein to the ACE receptor on the cells. And remember, those are present on lung cells, intestine, and so forth. If the antibody were to attach to the receptor binding domain up here in red, it might block the attachment of the spike protein to the ACE2 receptor, but in which case it would neutralize it. But if it attaches to this N-terminal domain over here depicted in green, it'll actually make the virus more efficient at attaching to an ACE2 receptor and getting its genetic material into the cell. Okay, so that's the third type. Now, the question for our purposes tonight is, can ADE occur? Remember, that's antibody-dependent enhancement. Can that occur with the messenger RNA vaccines? Yes, it can. Is there cause for concern? Which is the big question. And the answer to that is, I'm not sure. Which in another way of saying is, yes, there's cause for concern. Unless you're absolutely sure that this cannot happen, you certainly should be concerned about it. Many scientists believe this happened with the animal model used in the SARS I and MERS vaccine trials. Ferrets were used because their respiratory system is similar to humans. And they died at an alarming rate after getting the vaccine now compared to controls, the controlled ferret group, which did not get the vaccine. So why would the ferrets that got the vaccine die at a higher rate when exposed to the virus, SARS-1, than the ferrets that did not get the vaccine? And it's because of this ADE, we think. The ferrets that got the vaccine had antibodies produced to it that aided in the enhancement of the disease when those ferrets were exposed to the disease, okay? And with the mechanism I just showed you. it helped the phagocytic cells bring the virus inside the cell. And since they had not been neutralized, the SARS-1 virus had not been neutralized by the vaccine, it actually made the disease worse and the ferrets died. Okay. Another thing that is concerning to me a little bit is that we can't get up-to-date, accurate death counts from anywhere that I've found. I've just been unable to find them. Okay. Now let me just talk about dengue hemorrhagic fever, because it's a very, very good model for this phenomenon, ADE. There's 4 types of dengue viruses. And when you get a first infection by dengue with one of the 4 types, the antibodies that are formed are not strong or specific enough to neutralize an infection the second time you get dengue with a different type, okay? So let's say this is I'm using the same pictures here because I want to make sure you understand it exactly in the context of what I've already showed you, so I don't confuse you with too many different pictures. So here's a protein, just like the spike protein, that allows the dengue virus to get into the cell. And let's say that you were infected with dengue type A the first time, and those antibodies, attach to the antigens on the spike protein in unimportant places, such that the virus is not neutralized, okay? Remember, this is the type A antibody, and then the second time you get infected with dengue, maybe it was type B dengue, okay? It's one of the other 4 types. And when that happens, those antibodies are not going to do you any good. They're only going to serve to help the phagocytic cell bring the virus into the cell where it can hijack the machinery and make more copies of itself. And that's why dengue fever is much worse the second time you get it. Okay? That's a classic example of what happens with ADE, dengue fever. All right, now let's talk about it as it relates to SARS II, okay? I got these diagrams and the quotes on the next page from a paper from Cambridge University. And so I just put the title of it here in case anyone wants to look it up. But basically, here's what's going on with this. Normally, a virus will come up to a angiotensin converting enzyme, ACE2 receptor, as I've already explained in the primers on the previous video presentations, and allow its genetic material to come into the cell and go through translation and make all the parts of the virus, assemble them, and release, okay, to copy itself many times. With antibody-dependent enhancement, though, you don't need an ACE2 receptor to get into the cell. The phagocytic cell, in this case, it's a monocyte. It could be a dendritic cell, it could be a macrophage, etc. You don't need the ACE2 receptor because the opsonization of the antibodies attaching to the virus here make the virus more delectable, easier to phagocytize by the macrophage or monocyte. And of course, the viral material, the viral messenger RNA gets delivered to the cytoplasm, and it gets interacts with ribosomes and makes the proteins and makes more copies of itself, which get released. Okay, so with ADE, you don't need the ACE receptor. Now, there's been a lot of talk about what happened with SARS-1 and the ferrets. And I found this also in the same article, which is very, very important here, because it shows SARS-CoV-2, the protein list here, that is involved in ADE. And for SARS-CoV-2, the protein sequence is exactly the same as in SARS-1. Okay, now there are differences in the proteins in these two viruses, of course. There's a lot of commonality, but there's some differences. But as far as the ADE sequences, they're exactly the same. So maybe you could stop the video and convince yourself, you know, it goes LY. Let me get my glasses on, my heavens. Sorry. It goes LYQDVNC for SARS-2, and for SARS-1, it's the same. It goes LYQDVNC, exactly the same, even though some of the other proteins differ. But for as far as the ADE sequence, it's the same. So that's a little scary. And we already know that for SARS-1, we're pretty sure that the ferrets died because of this phenomenon. So that concerns me, certainly. Now let me read, and I blew it up. I should have read it off this one, but I re-pictured it here so that you can refer to this if you want as I read these quotes from the paper. It says the protein sequences responsible for ADE have been identified on the S protein, that's the spike protein. Importantly, sera from SARS-1 patients contain a mixture of IgG antibodies that both inhibit function and cause ADE. In other words, some of those antibodies will neutralize the virus and some will cause ADE because they can't neutralize the virus. All right? So they're different antibodies going against different epitopes or antigens on the spike protein, just like I showed you with dengue fever. All right. Similarly, vaccination, now we're talking about vaccination with recombinant S protein in animal models can elicit both neutralizing and ADE inducing IgG antibodies. So the vaccine can do that too. Even the presence of neutralizing antibodies can cause severe disease, including the cytokine storm, similar to that seen in dengue virus. Now, that's the mechanism number 2 for ADE. All right? It should be no surprise that a vaccine that delivers the entire spike protein is going to make lots of different kinds of antibodies, some to the receptor binding domain, which will neutralize the virus, and some to unimportant domains that will only serve to potentiate ADE. Okay, so this concerns me greatly, you know, because we're getting the messenger RNA for the entire spike protein. So if it makes antibodies that don't neutralize that virus, they're only going to cause ADE to occur and could make the next infection by that virus much worse. And I'm going to talk about that in the context of all these new variants that have come out, because remember how I talked about dengue fever having 4 types? Well, isn't that sort of what's happening now with SARS-2? We're getting the South African type or variant. We're getting the Great Britain type or variant. We're getting one from Brazil. So we're getting SARS-2 variants, which is like different types that are similar enough to call them SARS-2, but maybe the antibodies that we're making from the original vaccine, which is for last year's SARS-2, will not neutralize the South African variant fully, and it may cause ADE to occur. And certainly after several mutations have occurred, the SARS-2 is going to change enough to where the old vaccines produce antibodies that do not neutralize the virus anymore. And that at that point, will they cause ADE to occur if they're not causing it now from the new variants? So there's a lot of risk in the way I see this. Now, I'm going to bring on an expert in a second who's going to have a different opinion than me, which is nothing new. But let's see what he says in just a second, and then we'll discuss that. If I can bring the video up, I hope I can. Okay. I don't believe I've read the second paragraph yet, so let me read this. pre-existing anti-coronavirus IgG antibodies that cross-react with SARS II, including those against common and less pathogenic coronavirus strains, may increase the risk of ADE and the severity of COVID-19 disease. Now, so what they're saying there is they're talking about just IgG antibodies to pre-existing pre-existing coronaviruses that you've had before, maybe from the common cold. They're less pathogenic strains that maybe they cause the common cold instead of something much worse like COVID, okay? That's scary, isn't it? In other words, could COVID be ADE for a common cold virus? Think of it that way. In other words, we're having this horrible trouble with COVID, and the antibodies that we form to the common cold are similar enough to SARS II, but not specific and strong enough to neutralize it. You see what I mean? Okay, that's a little bit of speculation there, but that's sort of the way this thing's turning out a little bit. Let's read on. This is a well-described phenomenon for dengue fever where antibodies against one strain are a risk factor for severe disease during infection with another strain. That is exactly what happens in dengue fever, and it's very analogous to what is starting to happen here with SARS. Okay. Thus, high affinity anti-CoV IgG may be most effective not only in neutralizing CoV binding during infection, but also increase the risk for ADE. Okay, it's exactly what I just said. This is not a hypothetical concern. Clinical trials for dengue virus and respiratory syncytial virus, RSV, vaccines were halted when vaccinated subjects were found to have increased disease severity after viral infection. In other words, they were trying to make a vaccine for dengue fever, and they had to stop the trials. They had to stop the clinical trials because the people that were getting the vaccine got much sicker after the vaccine, just like the ferrets, okay? And in SARS 1, okay? Now, here is our expert, Dr. Offit. He is a world-renowned expert, and when I looked up tried to get some information on this, I saw that he has been consulted by scientific groups, public interest networks and stuff like Good Morning America and everything. The doctor has literally dozens and dozens of videos on YouTube talking about vaccines. And so I'm going to try to play this video now. I'm not sure I'll be able to pull it off, but I'm going to try to play the video of Dr. Offit talking about ADE and why he feels you don't need to be too concerned about it in SARS 2. And then I'm going to sort of analyze what he talks about as our final part of this video. So let me try to do this. I need to go up here and I need to share, new share. And then I need to go here, I think. Oh, bloody, what did I do? That is, I think maybe it's here. I don't know. It doesn't look right all of a sudden. Let me put my glasses on. Thought I had this, and it doesn't look like I have it anymore. It should be a regular YouTube thing. Please come up somewhere. Messenger, brother. It might be this one. I'm going to try this one. Yes. Okay. Let me see if this works now, you guys. I hope you can hear this. Let's see.
My name is Paul Offit from the Vaccine Education Center. One issue that's come up regarding the safety of coronavirus vaccines is, could they cause something called antibody-dependent enhancement? Now, this is something that's been seen with a virus called dengue virus, which is actually a very common virus in this world, although not common really in the continental United States. When there are 4 different types of dengue virus. When you're infected with one type of dengue virus, and then you recover, and then you're infected with a second type of dengue virus, sometimes what happens is people, because they've developed an antibody response to that first and really different type of dengue virus, when they're then exposed to a second type of dengue virus, actually have much worse disease than if they've never been infected with any type of dengue before. So people are worried that maybe with this virus, this SARS-CoV-2 virus, that could also be true here, because there was a suspicion from some of the animal model studies that were done with SARS-CoV-1 virus, which was a virus that sort of came up in 2002, but then in a couple of years was gone, that also might be true here. But there are a number of reasons why it's unlikely to happen, very unlikely to happen. First of all, is if you're infected with human coronavirus, and there are 4 different types of human coronavirus, and then you're infected with a second different type of human coronavirus, you don't have that phenomenon of so-called antibody-dependent enhancement, where you have a much worse second infection than first infection. Secondly, when you look at people, and these were studies done in the early 1990s that were experimentally challenged with human coronavirus, and then challenged again with human coronavirus, sort of under sort of the experimental conditions, also, you never saw antibody-dependent enhancement. Third, if you look at animals who are infected with SARS-CoV-2 virus, experimental animals, and then experimentally infected again with SARS-CoV-2 virus, there's not antibody-dependent enhancement. And similarly, if you look at animals that are given a SARS-CoV-2 vaccine and then challenged with the natural virus, the SARS-CoV-2 virus, they too don't develop antibody-dependent enhancement. And then lastly, and finally, with people who are infected with SARS-CoV-2, when they are given antibodies, so-called convalescent plasma, directed against SARS-CoV-2, they too don't develop antibody-dependent enhancement. So I think the concerns that were raised by what happened with SARS-1 are understandable, but we now have 5 separate pieces of evidence that suggest that is not going to be a problem with this virus or with this vaccine associated with this virus. Thank you.
Okay, let me go back to the other page now, because I do have more stuff to talk about here. Let me try to find this now. Good heavens. How do I get back to my thing? I'm sorry, you guys. I don't really know how to use this stuff yet. I believe it's this one. And I have to go to this, of course. Okay, I'm back to my presentation. Now, you heard what Dr. Offit said there. It contradicted very clearly the paragraphs that I just read from the Oxford paper, from the Cambridge paper. So this presentation today is a little bit more difficult to follow than some of my previous ones, but I just could not simplify it beyond this. So I urge you, if you're interested in this and you want to hear, you want to really understand this and see that there's a difference there from what Dr. Offit said and what the paper said, please go back and take a look at it. You might want to watch this twice. I certainly spent an hour or so looking at all of this and trying to figure out what I wanted to say about it. So let me continue on, but I urge you maybe to listen to Dr. Offit again after you've read those quotes under the very colorful graph that showed that the sequences for SARS-1 and 2 were the same. Okay, the ADE sequences. So let me continue on, and I have a few things to say about what Dr. Offit said also. I found some logical flaws in it. So let me talk about those now, because again, this gentleman, this doctor is an expert, and he has been an expert for many years. He must know much more about vaccines than me. Although when I listen to what he just said, I see a little bit of 2 + 2 = 5 in it. It doesn't make logical sense. And let me explain why. All right, so first, I just wanted to give you an example of, not an example, but just reiterate what happened in SARS-1. Ferrets got vaccinated in the trials. Ferrets died in greater numbers compared to the controls, which did not get vaccinated when they were exposed to the virus. And it was believed to be due to ADE, a la dengue fever. And this is what a ferret looks like here. They look a lot like minks. You've heard of minks before. Women like to wear mink stoles years ago. All right. Now I think this is the final slide. So let's talk about what Dr. Offit said. I transcribed what he said because, or at least transcribed it sort of in a slightly tighter summary, as accurately as I possibly could, because we need to look at very carefully at what he said. And please go back and listen to exactly what he said, maybe while you're looking at this slide, to make sure I didn't make a mistake, but I was pretty careful. So let's look at this, because some of the things he said to me are self-contradictory. So first he implied that SARS is not like dengue fever because dengue has 4 different types of virus, which are similar and not the same. But that's sort of exactly what's going on with SARS II, isn't it? With SARS II, we have the original one, if you want to call it that came from Wuhan. They call it COVID-19. Now we have the South African variant, the Great Britain variant, and the variant from Brazil. So they're similar enough to still be called SARS II, but they're different because the spike protein has mutated. So what concerns me is that we will be given a vaccine for antibodies to the spike protein of COVID-19, and it will not neutralize. It'll help a little, but it won't completely neutralize the spike protein on one of the new variants. And it will only succeed in helping ADE to occur, okay? Which would make the illness much worse the second time, okay? And it may not happen with the South African variant or the Great Britain variant or the Brazil variant, but there will be other variants that come along in the future because the spike protein mutates so much. And eventually, the antibodies that we made for COVID-19 are not going to work against the 2nd or 3rd or 4th generation variant. And that might be when ADE occurs, okay? So that's the first thing that I noticed, that Dr. Offit said something that didn't make a lot of sense to me. Okay, then he listed the 5 things. And here was number 1. If you're infected with one of the 4 coronaviruses and then with another, you don't get ADE. Okay, he said that. That was reason number 1. And is he sure about that? I mean, we've all been infected with the cold, and then this year, some people got infected with SARS II, and then they got really sick. Is it possible that SARS II was really antibody-dependent enhancement occurring before our eyes? In other words, we had the antibodies to one of the weak cold coronaviruses, and when we got this new thing in here, they didn't completely neutralize it, so it made the disease much worse. Now, I'm speculating here. I'll even call it wild speculation, so I don't want to spend any time on it at all. I'm just throwing that out there as a possibility that contradicts statement number 1. Then he said humans experimentally challenged with human coronaviruses and then challenged with human coronaviruses again did not get ADE. Of course, that's experimental. That's not in the real world. Then he said animals experimentally infected with SARS II and then infected again with SARS II did not develop ADE, okay? Then he said animals challenged with a vaccine and then challenged with the virus did not get ADE. That may well have been true in the recent trials with SARS II, but that is exactly what happened in SARS I when they did get ADE and all the ferrets died. Compared to or many, many more ferrets died in the vaccinated group than in the control group. So number 4 there may apply to SARS II, but it certainly would not apply to SARS I. And again, the ADE sequences are the same for the 2 viruses. Go back and look at that table with all the colors on it. So that concerns me a little bit. Also, I'm not really sure the extent to which animals were included in the trials this year. I haven't really seen the definitive answer on that. You know, I've heard rumors that they didn't do animal trials, they jumped right into human trials because they were in such a rush, in which case this wouldn't mean much to me because they didn't extensively test animals with the vaccine. But I may be wrong about that. Maybe there were plenty of animal trials. I just don't know. By the way, the human trials are ongoing. They're not going to be done until late 2022 or early 2023 for the Pfizer and Moderna vaccine. So the trials aren't even complete yet, and we're administering this vaccine to people. So in a way, they're being used to see what's going to happen in a way because the trials aren't finished yet. All right. Number 5. Now this one really bothered me because it kind of displayed a misunderstanding of ADE. So let's read it and let me tell you why I think that. It says people infected with SARS II. This is Dr. Offit's 5th reason why ADE is, quote, unlikely to happen. People infected with SARS II who then get antibodies by way of convalescent plasma don't get ADE. But of course they wouldn't get ADE because he's got something backwards there. You get ADE when you get the antibodies first and then you get the virus, not get the virus first and then the antibodies. That's not going to cause ADE. You need the virus to come in second when the antibodies are already there so the macrophage can phagocytize the virus and cause all the problems where it multiplies itself. Not giving antibodies after you've given the SARS II. So that just made no sense at all. Okay, now let me, and I don't want to be too critical of Dr. Offit here because, you know, he's not here to say, oh gosh, I made a mistake. I said something I shouldn't have said there, which I would assume he might say if he were here. So let's don't be too critical about that. But what he listed for his 5th reason certainly is not a reason. I'd love it if he would see this and contact me so I could interview him about this because this is very interesting, and I'm sure he's interested in this, and maybe we could get him to answer our questions. But let me move on now. So there is my presentation on ADE, which concerns me greatly, and incorporation of genetic material from a vaccine or a viral vector into our human genome, okay? The recipient's genome. There are other risk factors associated with getting these vaccines, which I may cover in yet another part. But I wanted to keep these things short because I tend to make them too long and people don't watch them as much as they would if they were shorter. So I've made about 25 videos now on COVID, the vaccine, and some other topics. And please put in my name and Truth in Science on the search bar of YouTube, and you'll get to go back and look at some of these presentations if you want. There's a lot of primers in there on immunology, how the vaccine works, how hydroxychloroquine works, how ivermectin works, vitamin D's role, and so forth. And if you're not up on it and you want to be, those things will teach you everything you need to know. So again, thank you very much for putting up with me, my wordiness, and I hope I've shed a light on some of the risks involved in getting the vaccines with tonight's presentation. And thank you for watching, and please comment in the comment section. I really learn a lot from what you guys point out to me and ask me to look at in future presentations. Thank you very much, and I'll see you down the road. Thank you.